All right, so today we're going to be talking about hallmarks of cancer. Hallmarks of Cancer is a paper that is so important in the world of oncology. Received tens of thousands of citations, and really, it's amazing because it's this conglomeration of the knowledge of oncology, and it has been published in 3 editions. So this paper was first written by Dr. Douglas Hanahan and Dr. Robert Weinberg, and it gave a foundational framework for understanding what makes a tumor a tumor. The identification of these properties have served as identifiers and signals for where research is heading, and it can serve a great entry point to be a great entry point for those who are entering this line of research. However, it has a relatively large barrier of entry with these papers, and I hope to make them accessible to the average viewer through this video by distilling the concepts discussed in this review to its basics while not losing the depth and specificity. So, I couldn't explain this paper better than Dr. Hanahan did in saying that the hallmarks of cancer were proposed as a set of functional capabilities acquired by human cells as they make their way from normalcy to neoplastic states. So these two doctors outlined these 6 hallmarks of cancer that they said is what characterized different tumor cells, and this is what they all had in common. It was later expanded to 8 based on observations until 2011, and was then expanded to 12 based on their work that was published in 2022. But over here, what they have published, the 6 hallmark capabilities as of the paper published in 2000, are self-sufficiency in growth signals, insensitivity to anti-growth signals, tissue invasion and metastasis, limitless replicative potential, sustained angiogenesis, and evading apoptosis. I will discuss all 6 of these features from the paper. Each in isolation isn't symbolic of much. However, they wreak chaos in the body when they act together. So I'll discuss the future of these papers in a more contemporary version in successive videos, but this should give the lay of the land in cancer research pre-2000. So the first part we'll be diving into is the self-sufficiency in growth signals, right? So it's basically this part. It's right at the top. So imagine you're at a castle, right? Let your imagination go wild. Imagine the cell represents a castle. These are the guards that are stationing it right here. Oh no, I must change the depth. So these are the guards that are stationed on the castle, and their purpose is to ensure that no one can enter and no one can exit. So just label these as the guards. So we also have messengers, as annotated over there, who travel from the outer walls of the king court and supplies that must enter and exit the complex. So just make an arrow and make this supplies. This is stuff such as food and all for the constituents who live there, and these food and resources carry out the growth. Now, imagine what happens in cancer is that the guards at the border of the castle are constantly receiving orders from the outside to expand or grow. So imagine that this is just one small constituency within a huge state. So it's receiving orders to consistently grow and expand. And these instructions are going from the guard through the messenger all the way to the king, and the king is carrying them out. So what's happening is that there's a constant supply of food and resources to promote this growth. However, this growth has a consequence. It's unpredictable. The structure, because of this rapid growth, the structural support in certain places might be weaker or stronger to the detriment of the kingdom. And remember, this is just one small constituency. If this grows way beyond its means, it's taking away space from the other kingdoms. And even though it might be good for this individual kingdom itself, it can lead to the detriment of the other kingdoms. So it might sound like you want the castle to grow, but this isn't the only castle in the kingdom, right? Because it's interrupting the growth of other castles and taking away space from other kingdoms destroys the balance. So this breaking of homeostatic balance is a key characteristic of cancer. So to explain what I just said in scientific terms, normal cells need mitogenic growth signals to transition from a resting state to an active proliferative replicative state. The transmembrane receptors, who I call the guards, so let's just mention these as T-transmembrane receptors. So these transmembrane receptors located at the membrane of the cell are constantly giving signals encouraging growth, and the signaling molecules within the cell are relaying this information. So the messenger is the signaling molecule. You might want to reduce the thickness of this one. Handwriting would be worse. So these are the signaling molecules. And what happens is that this is constantly going to the king, and the growth factors, which are basically the supplies. these are the growth factors, are constantly being supplied to facilitate this growth. And these growth factors aren't being, the growth factors we'll get into later because it's a key part of angiogenesis as well, which we are going to be talking about. It is a hallmark of cancer, as I said. So the extracellular matrix components, which is basically you can consider to be the walls of the castle, may reinforce or weaken the binding, and the cell adhesion from cell to cell, which is what allows the tissues to stay together, can also be weakened, resulting in these cancerous cells, these malignant cells, detaching and invading neighboring territories. So it's like there's a corrupt officer over here, like him, a corrupt officer right there, and he's flying off and going to this other kingdom that we have three ugly dots. He's going and he's going to corrupt that area further. It becomes a lot harder to deal with this when that happens, and that's called metastasis. But we'll get to it later. So to go more into detail of how this growth signal autonomy happens and how this is allowed to happen in cancer, so we have three of these different strategies. Now, cancer cells often produce their own growth factors, which leads to autocrine stimulation, which means basically means they produce their own growth factors. For example, one common example is glioblastomas and sarcomas, which is basically cancers of cells of mesenchymal origin. And mesenchymal tissues are connective tissues such as bones, muscles, fat, or nerves. So for glioblastomas, there's an overproduction of platelet-dependent growth factors, which is called PDGF. So PDGF, platelet-dependent growth factors, and this is a growth factor you'll hear about with great frequency. And for sarcomas, it's TGF alpha, which is transforming growth factor alpha, which is written as, let me just write that down, sarcomas, TGF alpha. Yeah, so it creates a self-sustaining loop of growth signals. And I can attach an image of what the differences are between these two growth factors. Give me a sec. Okay, so down there is a quick image of what the differences are between TGF alpha and PDGF. You don't necessarily need to know these to get the understanding of what it means. Maybe later, if you want to go into more detail, you can. So, growth factor receptor means tumors frequently overexpress growth factor receptors, such as epidermal growth factor receptor (EGFR) and HER2. This makes them hyper responsive to growth signals. In some cases, these receptors are altered to signal independently on their ligands. Ligands are basically receptors on the membranes of cells. So you can think of it this way. It's when you're overly receptive to ideas. It's like, I'm not sure how to explain it. It's like being open-minded, but to a detrimental effect, where you're picking up bad ideas from the surrounding people. It's basically like peer pressure, I guess you could say. So, and then the third part is the intracellular signaling pathways. And this is very interesting because this is the part of cancer that's pretty much like, it's like a map because you need to understand, because what happens in bio is a flowchart of events, how one chemical reaction leads to another, and it's a cascading sense of signals, which ultimately leads to a change in gene expression, which expresses the changes that are being made or expresses the changes that the signals are trying to make. So this is the most complex strategy, and it involves changing multiple intracellular pathways. There are many intracellular pathways, but one of the most common ones that you'll come across is the RAS protein. And the RAS protein continuously sends growth signals without external stimulation. So I want to talk a bit more about RAS because of how important it is, and it can serve as a cool example of how cellular pathways work. RAS proteins they act as traffic controllers at a busy intersection in the city, determining how cellular signals flow based on incoming cues. Similar to traffic lights changing from red to green based on sensors detecting cars, RAS proteins switch between active green and inactive red states. Let me just show an example of what different cellular pathways look like. You can consider the image in front of you to be a very detailed image of cellular pathways. So it's a cascading series of events, and RAS proteins basically can either inhibit or promote certain signals in response to signals binding to receptors on the cell membrane. So when green, RAS proteins provide RAS proteins guide cellular traffic down specific pathways, leading to responses like growth and survival. However, what happens in cancer is that a mutation in the RAS gene can keep these proteins stuck in the green state, which causes cells to grow uncontrolled, be like a malfunctioning traffic light. And this image over here is considered to be like a cellular integrated circuit. I don't understand it. There is a lot that I don't understand because there is a lot that's in it. But you can go through the individual parts of this. It's also in the paper, which I will give the link of. And you can look at the cool names that might seem interesting to you. For example, this Mad Max complex. That's just a fun name. You can go online and search what it does, and it can lead you to very interesting concepts within the topic of cancer. So, and RAS is somewhere. I'm not sure where, but it is somewhere. So, but cancer cells in cancer cells aren't individually responsible for this because sometimes tumor growth isn't solely driven by the cancer cells themselves, but also the surrounding normal cells. These bystander cells, such as fibroblasts, and fibroblasts are cells that are used in connective tissues, and they promote proteins like collagen and elastin, which form structural framework essential for tissue strength and elasticity. So you can, and collagen is a name you must have come across when talking about blood clotting, right? So that gives you an idea of what it does. And also endothelial cells. And endothelial cells are cells that line the surface of either blood vessels or lymphatic vessels, and they form a barrier between the blood or lymph and the surrounding tissues. These two types of cells can play crucial roles in tumor proliferation through the signaling that they provide. This communication within the tumor microenvironment is essential for cancer progression. And more will be realized, of course, because this paper was written in 2000. There's a lot more to explore within it. So normal cells within the tumor release growth signals that support the cancer cells, sometimes evolving to sustain this malignant growth. So if we have the cancer cells, we have the cancer cells that are basically encouraging these or prodding these fibroblasts and endothelial cells to promote more growth signals. And they're sending them back to the cancer cells, the growth signals, which allows the cancer to multiply and multiply and multiply. And then it's going to send more signals back. And then it's going to send more signals back. So it creates a chain reaction and loop that's very, very difficult to stop. So the autonomy in growth signaling is not just about the cancer cells, but also involves manipulating the surrounding normal cells to support tumor growth. This complex interplay is a hallmark of how tumors grow and proliferate.